Welcome to this explainer. When we look out at a field of incredibly high-yield crops or maybe a really genetically robust flock of sheep, we aren't just seeing magic out there, we're actually seeing the language of genetics hard at work. Today, we are going to decode its alphabet and its grammar to understand not just how life sustains itself, but how we can actually step in and actively shape it.
Okay, let's dive into this. Here is our roadmap for today. First, the blueprint of life. Second, the genetic alphabet. Third, genotype versus phenotype. And finally, number 4, the pioneers of genetics.
All right, Section 1, the blueprint of life, science versus application.
To truly grasp this field, we kind of have to split it into two distinct but deeply connected camps. On one side, we've got animal genetics. This is the fundamental science. It deals with the study of heredity and variation. Basically, it's the biological mechanics of exactly how characteristics are transferred from parents to their offspring. But then, on the other side, we have animal breeding. Now, this is where theory actually meets reality. Animal breeding is the active application of those genetic principles to improve economically important traits in domestic animals. Think about a dairy cow producing way more milk, a chicken laying more eggs, or beef cattle yielding more meat. Essentially, animal genetics gives us the rules of the game, and animal breeding is how we play that game to win superior merits and better resources.
Moving right along to Section 2, the genetic alphabet, building blocks of heredity.
We're going to run through this pretty quick to get the vocabulary down, starting with the trait. A trait is quite simply a characteristic, something like the color of a flower, or an inherited hair pattern that gets passed directly down through the genes from parents to offspring.
Now, driving those traits is the gene. As the smallest unit of inheritance, the gene is what controls exactly which trait is expressed. And crucially, genes always occur in pairs, one from each parent. At the microscopic level, these nucleotide sequences carry the exact instructions to construct proteins. You can think of them as the core letters of our genetic language.
And because genes come in pairs, they often have different versions. We call these alternative forms alleles. So if the gene dictates flower color, the alleles are the specific instructions for, say, red or white. And when these different alleles find themselves paired up in a single organism, a truly fascinating biological battle for dominance begins.
Now, what's really interesting about this biological battle for expression is how the dominant allele aggressively takes charge, essentially silences the other one. Always represented by an uppercase letter, the dominant allele completely dictates the outcome in a hybrid. Meanwhile, the normal, or recessive allele, which is represented by a lowercase letter, just recedes into the background. It physically manifests much, much less often.
To put that into perspective, let's think about how this actually looks. When an organism has two identical alleles, like, say, two dominant genes creating a pure red flower or a pure white one, we call that a homozygous genotype. They're purebreds. But when you combine one dominant and one recessive allele, you get a heterozygous genotype or a hybrid. This mixed genetic combination can lead to entirely different expressions, just like varied red and white patterns you might see in a hybrid flower.
Which brings us to Section 3, genotype versus phenotype, inside versus outside.
So the crucial point here is that there is a very distinct separation between the inside and the outside. The genotype is the internal genetic combination. It's that hidden code, the uppercase RR or the mixed RR that we just talked about. You can't just look at an animal and instantly know its genotype. The phenotype, however, is the physical feature that results from that genotype. It's the red or white color of the flower. It's the actual physical trait we can observe with our own eyes. Simply put, genotype is the blueprint and phenotype is the finished building.
That takes us to Section 4, the pioneers of genetics, the detectives of hereditary.
And this brilliantly illustrates the work of our first pioneer, the Austrian monk Gregor Mendel. Back in the mid-19th century, Mendel studied the segregation of heritable traits using pea plants. By carefully crossing them over and over, he noticed an absolute revolution. The results, like seed color for example, were always discrete. They were yellow or they were green. There was absolutely never a blended intermediate. This effectively proved that heredity is passed down in distinct, unbroken units, which we now know as alleles, rather than just blending together like two cans of paint.
Yet, Mendel's story is honestly one of science's great tragedies. He presented his incredibly precise mathematical patterns in 1865, but the scientific community just wasn't ready to understand them. Mendel died in 1884, completely unaware that he had changed biology forever. It actually wasn't until the 1890s that three separate scientists, Hugo de Vries, Karl Correns, and Erich von Tschermak, who were working on similar problems, rediscovered Mendel's laws. They suddenly realized he had solved the puzzle of inheritance decades before they even got started.
Now, around the exact same time Mendel was quietly growing peas, another pioneer, Charles Darwin, was looking at inheritance on a massive, global, evolutionary scale. Darwin gave us this incredibly famous concept, survival of the fittest. He realized that in nature, individuals of the same species are constantly competing for the exact same limited resources. We're talking food, water, space, and mates. In this brutally harsh competition, only the organisms best equipped to survive will actually live long enough to reproduce and pass their genetic traits on to the next generation.
Let's move on and see how this builds into the very foundation of modern breeding. We essentially have two powerful forces at work here. Natural selection is the blind, unguided hand of nature, naturally choosing the organisms best equipped to survive harsh conditions. Artificial selection, on the other hand, is where human ingenuity steps in. Instead of waiting for nature to choose, we deliberately select the highest performing males and females. We actively engineer the mating to produce superior offspring with the exact economic traits we desire. We are literally taking the tools of evolution into our own hands.
So what does this all mean for the future? We've journeyed all the way from the microscopic letters of the genetic alphabet to the massive global forces of natural and artificial selection. The language of life is incredibly complex, but its rules are undeniable. As our tools become sharper and our ability to read and write genotypes becomes faster than ever, we have to ask ourselves, how will our modern understanding of these ancient genetic principles continue to reshape the breeding and survival of species tomorrow? The blueprint of life is quite literally in our hands now. What will we build with it? Thank you so much for joining me on this explainer.